Okay, so thank you. I was meant to be sharing this session with Steve O'Brien, but he's actually had to leave, so I've got a few of his slides as well to present. So these are the studies that are currently available within the UK or have shut within the last few years. I'm going to tell you a little bit about the studies that are currently open, the CALLS study, Optic, and the SEAW1 study. I'm also going to tell you a bit about Spirit 2, Choices and Matchpoint, which are now closed and either reported or in follow-up, and then a little bit about Taster, which is a new study that will be coming along next year. And Professor Richard Clark is going to spend quite a bit of time on Destiny after me. So I'll start with Spirit 2. So I'm sure a number of you were in Spirit 2. It was a randomised study comparing dasatinib with imatinib in patients with newly diagnosed chronic myeloid leukemia. And this is the final analysis of the study. The study reported in March of this year. So this is the design of the study. It was for patients that were newly diagnosed with CML, so within 3 months of diagnosis. It recruited a total of 814 patients, and patients were split equally between imatinib, 400 milligrams daily, and dasatinib, 100 milligrams daily. This is looking at the molecular responses, so this is the major molecular response rate over the 5 years of the study. And we can see that dasatinib in the blue line is consistently superior to imatinib. So the MMR rate with dasatinib at 5 years was 83% compared with 63% for imatinib. However, as I think Professor O'Brien showed earlier, if we then try and relate that to overall survival, we see that actually it doesn't matter which arm you're on, if you're on imatinib or dasatinib, the overall survival is absolutely identical, as shown by the red lines and the blue lines completely overlapping each other. So summary from the Spirit 2 study, the PCR response is better with dasatinib, but we see no difference in event-free or overall survival. So although we're seeing faster and deeper responses with dasatinib, it looks like you will get there in the end with your imatinib, maybe with fewer side effects. What we did find with the Spirit 2 study was the pleural effusion rate was 36%, which is quite significant. And actually in patients over the age of 60 years receiving dasatinib, the rate of pleural effusion was around 50%. So again, that's a significant consideration in terms of side effects. The next study I want to talk about is called the Choices Study, and this is a study that was run through Glasgow by the late Professor Tessa Hollyoake, and again, that this study has reported this year. And it was for patients that had achieved at least a cytogenetic response, but still had detectable BCR-ABL by PCR. And it was adding in another drug called hydroxychloroquine, which is actually an anti-malarial drug and also used to treat rheumatoid conditions, but the hydroxychloroquine affects a process within the cells called autophagy, which is something whereby the cells autophagy stands for eat yourself. So what happens is when the cells are treated with a TKI, because the TKI shuts off essential pathways within the cell and the cell's metabolism, the cell then turns to other pathways to try and survive, and that process is called autophagy, and this allows the cancer cells to survive longer. And hydroxychloroquine and drugs like hydroxychloroquine are able to inhibit this process of autophagy, which then increases the chances of the cancer cells from dying. And cancer cells are much more dependent on autophagy than normal cells, hence the reason for combining the imatinib with the hydroxychloroquine. And these graphs just show autophagy in action, if you like. So in these two figures at the top show CML cells. The one on the left is untreated, and you can see that there's not a lot of green dots on the left. When you treat with dasatinib, it induces autophagy, and these the big green dots are these areas of autophagy where the cell is gradually eating itself to try and survive. So the Choices study was carried out across a number of sites in the UK and also sites in France and Germany. And it recruited a total of 63 patients, and it was a straight one-to-one randomisation of patients continuing on imatinib on their own or the imatinib with the combination of hydroxychloroquine. And they stayed on the combination for a year and then went back on to imatinib on its own. And the primary endpoint for the study was a half-log reduction in BCR-ABL levels. So it was an improvement in the BCR-ABL level was what we were looking for with this study. So what we found in terms of molecular response was actually we saw a very slight improvement with the combination with hydroxychloroquine, but it didn't reach what we would term statistical significance, so it wasn't substantially better than the imatinib on its own. In terms of safety for the study, we had 17 adverse events reported. The majority were actually in the imatinib arm, and we don't believe were related to therapy. If we look in terms of tolerability, we actually see that for most patients, the combination of imatinib and hydroxychloroquine produced some side effects of diarrhea, and this was worse than with imatinib on its own. In terms of musculoskeletal side effects, interestingly, we didn't see any musculoskeletal side effects on the combination, and we know that hydroxychloroquine can be used to treat diseases such as rheumatoid arthritis, so that was quite interesting. But there was no significant differences in terms of other significant differences other than the ones that are shown here, so worse diarrhea with the combination and better musculoskeletal side effects. So the conclusions from this study, this was the largest autophagy inhibition trial run in a malignancy and the first in hematological cancer in the world. It did demonstrate tolerability of the combination. There was a trend towards increased molecular response with the combination, but this failed to reach statistical significance. The numbers in the study, however, were small. There was only a total of 63 patients in the study, and there was quite a lot of dropout from the study with side effects and with increased clinic visits. We don't know for sure if we go back and look at patients that were in the study in 5 years' time, whether we'll see a longer-term improvement, but we didn't see an improvement that was significant in the shorter term with the combination. And it may be worth revisiting this in the future. There are other autophagy inhibitors in clinical development that may be suitable for future studies. So this is kind of set to one side at the moment, but something we would consider again in the future. The next study I wanted to talk about was Matchpoint. This is a study for patients with blast phase CML and has recently closed to recruitment. So it was a phase 1 study combining ponatinib with intensive chemotherapy. It was for patients with either lymphoid or myeloid blast crisis. It could be patients presenting with blast phase or patients that had developed blast phase while on treatment. We recruited a total of 17 patients, and these patients are all now in follow-up. So the patients received 2 cycles of chemotherapy with ponatinib. At the end of that, patients either had an allogeneic bone marrow transplant or they went on to ponatinib maintenance. And the final results of this study will be available next year. I'm going to move on now to the studies that are currently open within our portfolio. The first is the CALLS study. This is a study for patients with CML and Philadelphia chromosome positive ALL. It's open in around 27 sites across the UK, and so far there's been about 50 patients enrolled, and the aim is to open 40 sites and recruit 400 patients across the UK. The main goal of the study is to determine the frequency of BCR-ABL kinase domain mutations in patients on TKI treatment using 2 different methods. Using the standard approach, which is Sanger sequencing, which has a sensitivity of down to about 20%. So you actually need to have quite a lot of the mutation there to be able to detect it. And we're comparing the standard method of detection of the mutations with something called next generation sequencing, which is using very sensitive DNA techniques to identify these mutations at very low levels down to 3%. The main eligibility criteria for this study is that all patients should have at least a BCR-ABL level of 0.1%, so they shouldn't be in MMR, they need to have a level higher than that, and they need to show criteria that don't fulfil the current ELN guidelines for optimal response. The second study is the Optic Study. This is a phase 2 study. It's aiming to recruit 450 patients worldwide, and we have a number of sites in the UK. Glasgow is a site, the Hammersmith is a site. I think there are a few other sites. Liverpool's a site. I'm not sure where else in the UK is a site. The main goal of the study is to assess how effective ponatinib is when administered at 3 different starting doses, 45 milligrams, 30 milligrams, and 15 milligrams daily in patients with chronic phase CML who are resistant to at least 2 TKIs. The main inclusion and exclusion criteria, so patients need to have chronic phase CML resistant to 2 TKIs and not have had prior treatment with ponatinib. And the thinking behind this study is that we know that there's quite a high rate of cardiovascular events in patients who have started on ponatinib at a dose of 45 milligrams. So we're looking to see if starting at a lower dose of 30 milligrams or 15 milligrams, you get an equivalent response in terms of how well the drug works, but you get a reduction in the problems with the heart and the blood vessels. The next study that's open is the SEAW1 001A2301 study. This is a phase 3 study looking to recruit 222 patients worldwide. It's open at 5 sites across the UK, Hammersmith, Cardiff, Oxford, Liverpool and Glasgow. And it's comparing the new ABL inhibitor asciminib, which works slightly differently to the standard TKIs such as imatinib and dasatinib. It binds to a different part of the BCR-ABL molecule, so it's not so susceptible to the kinase domain mutations, and it has a slightly different side effect profile. So this is being compared with bosutinib, and for every 2 patients that get asciminib, 1 patient will get bosutinib. And the main goals of the study are to compare the rates of major molecular remission at various time points with the 2 different drugs. The main inclusion and exclusion criteria are patients need to have chronic phase CML, need to have failed a minimum of 2 prior BCR-ABL kinase inhibitors, and they need to have a BCR-ABL level above 1% when they're screened for the study. And they can't have had prior therapy with either the ABL001, the asciminib, or bosutinib, which is proving to be a bit of a challenge because many patients in the UK have actually already had bosutinib by the time this study had opened. The final study I want to briefly mention is the TASTER study. So this is a study that has been funded this year by Cancer Research UK, and this is a study that we're hoping to be able to open next year. This is a study for patients that have failed all available TKIs. It's the first study of its kind funded by CR UK, and it's using a combination, with the clinical trial, we're doing a range of different laboratory assays to try and determine which treatment might be most suitable for which patients in terms of adding a drug onto the TKI that the patient is taking to try and get a better response. So we're going to take patients with chronic and accelerated phase CML. They need to be an ELN failure at the various time points and not a transplant candidate. We'll be looking to recruit 34 patients per arm from 15 sites across the UK, and the study will recruit over 6 years. At the moment, we're hoping to be able to open 3 arms of the study next year, a TKI-only control arm, a TKI with EZH2 inhibitor arm, which is a drug which targets how the sort of genetic control of the cells is affected, and a drug that targets the HDM2 inhibitor, which again affects protein regulation and cell death pathways. We have other arms that we hope to open a year or 2 further down the line, but those drugs are in earlier preclinical development, and we're waiting to get the results from early phase clinical trials in solid tumours to know that those drugs are safe to combine with TKIs. So what we're looking for is a well-tolerated second agent that also induces a half-log reduction in BCR-ABL by 6 months, and that this is durable over time. And what we're able to do with this study as well, for those patients that are in the control arm, or those patients that don't respond to the first small molecule, the first new drug that they get, they're able to switch at the first endpoint at 32 weeks, either onto the opposite treatment arm, or they'll be randomised onto one of the other treatment arms if they're on the control arm. So we're really excited about this study. It's very challenging to set up, but we hope it'll be open across the UK in a little over 6 months' time. And with that, I'm going to hand over to Joe, and we'll take questions at the end.